Hello everybody, and thank you for watching another video on qualitative and quantitative analysis of pharmaceutical dosage forms. In this slideshow, I'm going to talk about three different excipients: Ceteth 20, ceteareth 20, and cetomacrogol 1000. This presentation is useful for people who are doing reverse engineering of pharmaceutical dosage forms or cosmetic products. The Ceteth 20 and ceteareth 20 are non-ionic surfactants that have hydrophilic polyethylene oxide attached to fatty alcohols. Now, in case of these excipients, the polyethylene oxide is PEG 20 and the fatty alcohol is cetyl and stearyl alcohol. Ceteth 20 has a commercial name of Brij 58, which is PEG 20 attached to cetyl alcohol only. On the other hand, ceteareth 20 with the commercial name of Eumulgin B2 is a PEG 20 which is attached to cetyl and stearyl alcohol. Now, depending on who manufactures this material, you could have different ratio of cetyl and stearyl alcohol in the excipient. Or maybe the manufacturer will offer different grade of the ceteareth 20. Cetomacrogol 1000 is a trade name for ceteareth 20, although there are sometimes confusion between if the cetomacrogol 1000 is ceteareth 20 or Ceteth 20. There is a monograph for these excipients in the European Pharmacopoeia, and it is under macrogol cytostearyl ether. And this monograph describes excipients that has macrogol, which is another name for the PEG, attached to cytostearyl alcohol, which is cetyl and stearyl alcohol. And the PEG or macrogol could be PEG 2 up to PEG 33. So this monograph covers a wide range of different Ceteth or Ceteth 20 or ceteareth 20 type excipients. Also, there are different grade of commercial grade of this material available. We have Ceteth 2 up to ceteareth 100. Now, in case of ceteareth 2, it is a PEG 2 attached to cetyl and stearyl alcohol, and in case of ceteareth 100, we have a PEG 100 attached to cetyl and stearyl alcohol. So there are different grade of these excipients available in the market. In the FDA IAD database, there are 7 entries for Ceteth and ceteareth type excipients. So we have Ceteth 2, Ceteth 10, Ceteth 20, Ceteth 23, and we have ceteareth 12, 15, and 30. They are all used in the topical dosage forms, and they are creams, lotions, foam, used in solution and tampon. The maximum allowed limit present in the dosage form is listed here, along with the CAS number and UNII code number. For the qualitative and quantitative analysis, I'm going to talk about 4 different methods: HPLC with RI with and UV detector, HPLC with ELSD detector, colorimetric method that uses UV-Vis equipment, and GC technique. The first method is the HPLC with the RI and or UV detector. If you have the both detector attached to one HPLC, you can even do in parallel. It uses a mixed mode column And the detector in the case of UV is detection at 200 nanometer or 205, 210, or the RI detector. The mobile phase is the acetonitrile water, and which has a lot of acetonitrile content, and method is gradient. And this is the chromatogram of the pharmaceutical dosage form that has Ceteth 20 in it. And the Ceteth 20 peak comes very early at 2 minutes. And this formulation, particle formulation, had cetyl and a cetyl alcohol that shows at the peak around 3.5 or and 5.7. Now, in case of Ceteth 20, we see that Ceteth 20 has two peaks. The first one is the same peak that we saw in the Ceteth 20, that is peak 20 attached to cetyl alcohol. And the second peak in Ceteth 20 is a peak 20 attached to cetyl alcohol. And in this chromatography, we see that there is a coelution with the cetyl alcohol, free cetyl alcohol, and peak 20 sterile alcohol peak. So this method, isocratic method, is you can use it for quantitation of Ceteth 20, but it is not entirely suitable for quantitation of Ceteth 20. And also, you could do the grade determination. You can easily tell if you have a Ceteth 20 or Ceteth 20 in the formulation. The next method is HPLC ELSD detector. Now, in this case, the mobile phase A is acetonitrile water with TFA, and mobile phase B is methanol. Detector is ELSD, column is a C18. And this is the chromatogram of a pharmaceutical dosage form that has Ceteth 20 in it. And the Ceteth 20 peak, which is a peak 20 cetyl alcohol, is well separated from other excipients in the formulation, and we see a peak around near 18 minutes. Now, the next slide is the formulation that has Ceteth 20 in the formulation. And again, we see two peaks for Ceteth 20, and they are both well separated from other excipients in the formulation. And the first peak is peak 20 cetyl alcohol, excuse me, and the second peak is a peak 20 acetyl alcohol. So ELSD HPLC technique, we can do quantitation of these two excipients, and we can do grade determination easily. So it is better, in my case, is a better method for quantitation purpose when you have cetyl and acetyl alcohol in the formulation. If you don't, maybe both methods will work. The third technique is a colorimetric method, which is the USB method for assay determination of ploxylene. Now, ploxylene is a molecule that has this polyether chain in it, and it has a similar structure to PEG. And in this technique, you are going to make a complex of cobalt with this polyether chain, and it is all the reagent, the techniques, and everything is well described in the USB monograph. So in this technique, you will make a linearity solution, and you do the quantitation at 630 nanometer. And as I said before, this method will work with any molecule that has polyethylene oxide chain or polyether chain. It will work. It's not just for this particular molecule, ploxylene. It will work with the Ceteth 20, Ceteth 20, and all of them. The last technique is the GC method, and in this technique, then you need to do base hydrolysis, and you have to break off the cetyl and the cetyl alcohol into free cetyl and sterile. And then you do the GC quantitation. The method is for the cetyl alcohol is available, and it's very easy to develop. And then you do the amount determination. In this method, you could get information on amount for sure and grade to some extent, meaning that you will get information about the ratio of cetyl and acetyl alcohol present in the formulation, but you wouldn't be able to tell if it's, let's say it is CETA20 versus CETA30 or CETA100. That information you will not get. So I talked about 4 different techniques. The first one was HPLC with RI and a UV. You can do amount determination and grade determination with the first method, but as we showed you the chromatogram, the method development could be very challenging. Any changes or variation in your isocratic will lead to peak broadening, and in some cases you will not even recognize where the peak go. So it is challenging. Colorimetric method, it is only give you amount, but it usually works, and it works for all the molecules that contain PEG. But if you have other excipient that has polyether oxide chain in it, then you will have major interference. Let's say in your formulation you have both CETA20 and CETA50 or CETA60. Then you will not be able to do quantitation for CETA20 only. You will only get information for combined. HPLC ELSD detector, it gives you amount and grade, and in my opinion it is less challenging to develop a method. And the last one is a GC FID detector that gives you amount only and also to some extent grade, as I talked before. And unfortunately, if you have, in case of GC, if you have free cetyl and acetyl alcohol in the formulation, this will not work for you. The references are many, and you can search the website or companies that manufacture the columns, and you will get a lot of methods that they have published already. Also, the best reference would be USP/NF and AP. Thank you very much for watching this video, and have a great time.